Good morning, everyone. Thank you so much for joining us for this Enrich Biosystems webinar. My name is Jenna Gallegos, and I will be your moderator today. You are going to be learning about an integrated high-throughput platform for functional profiling of myeloid cells in models of pancreatic cancer. So without further ado, I will introduce our speaker, Dr. Jin Ho. Dr. Ho is a physician scientist at Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins. He focuses on cancer immunology and tumor microenvironment research. Dr. Ho received his BS and MS in biomedical engineering from the University of California, Los Angeles, and his MD from Wayne State University School of Medicine. Dr. Ho's overarching research interest is to develop novel immunotherapeutic strategies, even against immune-resistant cancers, by first understanding the immune response to current therapies. Without further ado, I will hand it over to Dr. Ho.
Great. Good morning, everyone. Let me just share my screen now. Okay, so it's really my pleasure to provide this seminar, and it's to showcase an integrated high-throughput platform. And the main point is so that we can phenotype myeloid cells within models of pancreatic cancer in a high-parameter fashion, but also obtain functional information at the same time. And so we call this a method that we have developed define direct evaluation of functional immune networks via ex vivo co-culture. The main problem and the setting in which we're doing our work that we're trying to address is this immunotherapy treatment gap. We all know that there has been remarkable progress in the field. It is now a mainstay for many cancer treatments and an option for majority of patients diagnosed with cancer. But if you look at this chart, where the y-axis is the percent of patients undergoing some immunotherapy with response, most of the cancer types still fall below the 30% response line. And this particular plot is in the context of a doublet immunotherapy, a checkpoint inhibitor against PD-1 or PD-L1 plus CTLA-4. And here at the very bottom left corner, a very lethal cancer, pancreatic ductal adenocarcinoma or PDAC, you can see has about 0% response. So no response to even a doublet checkpoint inhibitor. So we have a lot of work to do. And why is immunotherapy failing? And one of the major reasons are barriers within the tumor microenvironment. We know that the immune response is taking place. Even against this relatively non-inflamed tumor, we still have inflammatory cells. And they are actually barricading or suppressing the immune cells that are responsible for killing cancer, T cells, from doing their job. And one of the predominant features within the tumor immune microenvironment are these myeloid cells. It is a very complex and a far-reaching problem throughout many different cancer types. And here I'm just showing you within the context of PDAC that multiple studies that we have done in our group, and many others have shown this as well, that whether or not we give it some sort of an immunotherapy, and these are vaccine modalities, or standard of care, chemotherapy or radiation, all of the clinical outcomes are poorly associated when the samples have high content of myeloid cells. So this is an indication that they're doing something negative to immune system, and we have been accumulating in this knowledge for quite some time now. And how much do we really know about these individual myeloid cells and the complexity to them? Well, we're in really an insurgence of omics data, lots of so-called atlasing efforts, and especially in the recent years, we have been able to undertake amazing exploratory phenotyping and description of many, many cells within the microenvironment, and that includes myeloid cells. And so we've really built this literature and understanding with deep phenotyping of these cells. And as we do this, of course, we get to discover, we get to learn a ton about these and have a correlative understanding of all these cell types. The problem in the field, just a little bit more background, is that we are still some distance away from actually having myeloid reprogramming agents. We actually don't have a FDA-approved option to reprogram myeloid cells and really promote the immune system against cancer effectively and safely. So why is that? Well, despite all of these discovery process, we have this nomination bottleneck. We can discover, 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 but which of these targets actually give us a very good chance of perturbing the myeloid compartment to get us the immune response we want. And that requires functional insight. And so a lot of these omics and atlases have been correlative, and it really is limited in terms of functional insight. We also know that single cell sequencing, there's a lot of cost burden. And when we have cost burden, we have to then limit how many samples in what context that we're actually profiling. So the scope of insight is also limited. We usually look at one sample, and then that really refers to one clinical state that is being phenotyped. And so, can we actually perturb these? And can we add all these variables into this? And so that gets very limited. So, do we really know what we're looking at? And then also in single cell sequencing based profiling approaches, they're all RNA based, first of all. And then the protein level information is yet another layer of validation needed. And not only that, some of these methodologies require dissociation methods that will then lead to loss of certain subsets. So we have gaps in our understanding of certain subsets. And currently, this was reviewed some years ago, but our list remains short and pretty familiar at that. We have some strategies that we have tried in the field and to really limited clinical success and safety trade-offs. And so we have to do a lot more than this. And so this is just a schematic to the problem that we're trying to solve here. We have a lot of marker discovery and correlations. Then there is this bottlenecking towards then being able to nominate certain targets for mechanistic validation and then translational efforts thereafter. So there's a lot of time and labor expended to get to this point even. Whereas if we have a method that can help us to understand the functional implications right off the bat, along with discovery, then we may be able to speed up the discovery plus functional insights and then get to a higher rate of developing translatable strategies. And that's the goal of today's talk. Two parts that I'll be sharing with you is one, in PDAC, there are specific challenges with the tissue. And so we're optimizing dissociation. And then the second part is to then demonstrate the development and the use of our define method. So what do we do initially? As I mentioned, one of the problems is to get out the cells we want to actually profile. If we don't have the cells in the samples in the first place, it'll be very challenging to learn about them. And so to maximize our chances of getting the samples to contain the cells that we want to learn, we want to optimize dissociation. And pancreatic tissues are subject to autodigestion problems, a lot of enzymes that can lead to that. And so we have compared three different strategies that are used in the literature, commercially available, and also optimized in lab. And we have identified a way in particular to use collagenase and disphase to then iteratively spin down and collect the supernatants with gentle spins so that we can collect the tumor microenvironment cells without destroying them. And this is a panel of antibodies that we have used and optimized and validated using samples to phenotype myeloid, the compartment, to the best of our knowledge. So the selection of markers, and as you'll see later, this is for a mass cytometry based technique. And a lot of these antibodies are stemming from what's already known in the literature that's involved in anti-tumor or pro-tumor immune responses, but also just in general function of myeloid cells. We have some subtyping markers as well as functional state markers that denote different functions of these myeloid cells. So here it's just a direct head-to-head comparison of an optimized protocol so that we can yield a lot of cells out of PDAC tumors. You can see that we've compared three different strategies. One is a commercially available digestion kit for tumors that are either coupled or not coupled with a Percol enrichment, a density gradient enrichment of these cells. And what you can see is that one of the methods for pancreatic samples, although not a problem at all for other tissue types, For PDAC samples, they have really lost to near complete loss of some of the myeloid subtypes. So you can see that in three different tumors, there's really no yield for some of these cell types. And then when we get onto different strategies, really the most optimized way of dissociating these tumors to get at the cells that we want, we're really benefiting the yield of dendritic cells and Lysis G positive myeloid cells or Lysis C positive myeloid cells. And these are big increases compared to some of the standard or commercially available workflows. So now that we have a optimized way of getting all the cells we want as much as possible, then we couple this to a platform that will allow us to get out the functional information. And what have we done? Well, we have leveraged two hardware solutions here. One is through the Enrich Biosystems, the TROVO system, and then the other is with Standard BioTools, is a mass cytometry platform. And what are we doing? Well, we are dissociating these tumor cells. And then we're taking advantage of Trovo's ability to then print these microwells, thousands and thousands of these microwells onto these chamber slides. Of course, we could do it with microwells, but we're using a chamber slide for ease of then integration into imaging using a mass cytometer here. And then using these thousands of microwells, we are then setting up an assay of tumor kill. So these are PDAC cells, KP, so KRAS and P53 driven tumor cells that are expressing OVA. This is a very specific and strong immunogen. And then partnered with OT1 T cells. These are T cells, CD8 T cells that are able to recognize OVA epitope, sync-fecal, and then lead to its cytotoxic action. So we've set this tumor kill assay up, and then we then add these dissociated single cells onto these wells. And we do this in a stochastic manner where these are just a mixture of isolated cells that then are randomly able to distribute across all these microwells. And then we have distinct mixtures of myeloid cells. And then once we are imaging them throughout time to get the tumor kill data for every microwell that it has that can be tracked using barcodes, we also stain the entire slide with cocktail of antibodies that I've shown earlier to be able to phenotype the myeloid cells. And then we can phenotype every individual microwell and all the single cells within at the protein level. And so this is the overall workflow, taking advantage of the TROVO system and then the Hyperion system to be able to phenotype every single microwell and the contents therein, plus the tumor kill data, so that we can understand exactly what functional implications each of the phenotypes have. So this is really led by Nicole Gross, Dr. Gross, who's now finished her PhD in my lab. So the key components, again, KPOG plus OT1 and PDAC-derived CD111B positive myeloid cells. A constant density of cancer cells and T cells, of course, and then we add in these random mixtures of myeloid cells. So this is just a verification of the platform where we can see clearly the tumor kill. These cancer cells have a lot of OVA, epitope, sync-fecal expressed on them through the MHC molecule. You can see this population sticking out here in the presence of interferon gamma. And in the presence of interferon gamma, we set up the culture to really ensure tumor kill. And you can see that with the OT1 and plus interferon gamma, we were able to verify that that active killing is in process using the TROVO system. By the way, the KPOG1 cells also have GFP expressed within them so that we can track these cancer cells easily using a built-in microscope within the TROVO system. And then here is what happens on average. So we add the tumor mixtures of myeloid cells, so PDAC-derived myeloid cells, and you can see that when you add that on top of the OT1, the killing is suppressed. So on average, we can verify, as you would expect, that the mixture of myeloid cells on top of the T cells really lead to less kill. And we have hundreds and thousands of microwells with these mixtures, and we can look at the cancer kill across all of these microwells. And you can see that we can stratify them in best killing and worst killing microwells, and we can also track each of the wells over time. So what have we done with this printed microwells and all the cell mixtures within them? Using mass cytometry, we were able to get out protein level And since these are essentially antibodies that have been validated for flow cytometry as well, these are pretty high fidelity readouts. And we can get all these cells within the microwells at single cell resolution. So these are 1 micron imaging readout of every barcoded microwell. And so you can make out certain key phenotypes. So these are myeloid and T cells mixed in. You have also granzyme B positive cells or CTLA4 positive cells mixed in, as well as PD-1. These are checkpoint molecules as well as activation molecule. And then here we can track some of the myeloid-derived suppressor cell markers, CD262 or a granulocyte marker, and alternatively activated or M2-like macrophage markers, and so on. So we can get, these are just multicolor representations, but 40 plus parameters per cell and tracked for every microwell. And these are then clustered. We can take all of these images and single cell data and cluster them into specific phenotypes that we can recover. Here, across the worst killing and best killing cells, the density of CD8 positive T cells really is not a factor. And that makes sense because these have been normalized and controlled, and when we were even adding it in the first place. Now, when you look at the myeloid cells, however, one of the abundances for a subset, which is myeloid-derived suppressor cell, is an indicator of best killing versus worst killing. So, with a higher density of these MDSCs within these wells, as you would expect, it is associated with worse killing. And what's interesting is then we can take this 50 parameter panel of information and look at the functional states within each of these cell types. So this is a expression of a given marker at a per cell level. And we stratify this information again, best killing and worst killing. So you can see that CSF1R expression, which we have now in the field been interested in for quite some time. It's been established as a marker to target the myeloid compartment, especially the macrophages to hopefully yield anti-tumor responses. Higher CSF1R expression on TAMs or other myeloid cells and DCs. This is not otherwise specified. DCs, dendritic cells. So higher expression of CSF1R is actually identified in worse killing co-cultures. Higher levels of PDL1, very well established. We have therapies that go against this and actually work in the clinic. This is a nice validation to see higher PDL1 expression is also associated with worse killing. And then we have some here, right, and here higher expression of INOS, for example, within the MDSCs is actually associated with better killing. And this has also been shown in the literature that higher INOS leads to a microenvironment that is conducive to yielding cell kill. So this is what we're seeing here. And less sort of established in the literature are the CD262 and Fc gamma receptor. Now, we all know these Fc gamma receptors are very important in modulating the function of myeloid cells in taking antibody-mediated or opsonizing certain molecules or foreign bodies. So that expression, this is Fc gamma receptor 2b, is also identified in worse killing co-cultures. It's not really established well in the anti-tumor immune literature. And then higher expression on T cells of CTLA4, as we would expect, is associated with worse killing. And then, so what else can we do? Well, we can take the entire slide. That's a very high resolution phenotyping of microwells. And then we can also validate further using a whole slide imaging capability of the Hyperion system. And this is done at a low resolution, so it's very high throughput. The entire slide is captured in about 3 to 4 hours. And you can get out many microwells that are then phenotyped in a similar way, that you are clustering all of this data. And in a large scale analysis, we're able to recapitulate these markers. So high CSF1R expression on TAMs, DCs, and other myeloid cells associated with significantly more higher killing, and an INOS, I'm sorry, worse killing, and INOS with higher killing, and Fc gamma receptor 2b and TAMs associated with worse killing again. So now we have a larger scale validation of our high resolution data. And so what kind of insights then does that lead to? Well, directly what we're able to do is then take this information and inform some of the in vivo model efforts that's been going on in the field, right? We have many preclinical studies that use a variety of models, either subcutaneous tumors, liver tumors, or pancreas. And we often extrapolate these across, and this risks conflating their interpretations. And so this is important, especially when we're looking at the immune system, because there are tissue site-specific differences, and that can't be ignored. And so to directly compare using my lab's ability to profile the immune microenvironment within the tumors, we just simply take these three tumor models, PDAC tumor models, and then run my cytometry. We're able to see that pancreatic orthotopic tumor models have the lowest density of T cells. We're able to see within the subclusters of macrophages, certain subclusters are enriched within the PDAC. For example, you have this enrichment of these TAM1 is arbitrarily called a TAM1, and those are elevated with expression of CSF1R, Fc gamma receptor 2, live 1, PDL1 compared to other TAMs, arginase 1. And so these are all markers that associate with immunosuppressive capacity as we have seen in the defined setup. And that's enriched within the orthotopic pancreas tumors. Whereas these more inflammatory TAMs, we have iNOS and CD80 expression. These are more enriched within the subcutaneous tumors. So we can already have the functional data informing what kind of studies that we're doing in the lab all the time to get at the immunotherapy responses. So this is already a powerful tool that we can apply. And here, just very interestingly, these Fc gamma receptor 1 and 2, these are differential expression of the subtypes of these Fc gamma receptors. You can see that high expression of Fc gamma receptor 2 in these myeloid subsets, so macrophage subclusters, as well as MDSCs. And you can see that they're highly expressed more so in the liver and the pancreas tumors rather than the subQ tumors. And then the opposite true, these are more the stimulatory or activating Fc gamma receptors. These are more expressed in the subcutaneous tumors. So again, we can see that these functional information that we have gained from this combined phenotyping plus functional readout, actual cancer cell kill in a very high throughput manner, then already allow us to interpret these model differences, which would have just remained correlative otherwise. So just some take-home points and extended opportunities from what we have demonstrated today. So define enables high throughput marker discovery directly coupled with functional insights at the single cell level. Each run requires only a few mouse tumors. It is very cost effective. I just want to emphasize this, seeing that our field has really been able to do amazing things, but a lot of the methodologies still remain cost prohibitive. So we can compare that to how this run some of the runs that I've shown you really used hundreds of dollars. And so we can decide to run more runs without much cost burden relatively, and biological variability can be increased without cost burden, and we can pull more distinct mouse tumors. We can just perturb the co-cultures directly with siRNA, small molecule inhibitor, cytokines, biologics. You can imagine the functional implications that we can really screen in a high throughput fashion is enormous. And other TME mixtures can also be applied. For example, fibroblasts doesn't have to be just the myeloid cells. Readouts other than cancer cell kill can be adopted. So not just cancer cell kill as the initial setup. We've done that with KPOG1s and CD8 T cells as the main functional readout, but we can do this easily for other platforms or other systems, biological systems, for example, T cell activation that can be read out fluorescently directly within the TROVO system. Tumors can be modulated in vivo first, right? Vaccines, checkpoint therapies, or innate immune modulators, and then we can determine the functional consequences thereafter within the defined system or co-cultural systems, right? So we can get high parameter, again, rich with phenotyping information that is then coupled with some functional readout. And then we can think of combining with non-terminal platforms. As long as the imaging analysis workflow is worked out, we can then also take advantage of TROVO system where each microwell can be actually enriched and sequenced. And so if we're not forced to ablate these cells, which really is a requirement for the mass cytometry technique, we can do further applications. And then finally, just want to end with this point, prioritization, a platform like this, as opposed to simply omics, if I could humbly say, would accelerate target nomination and translation of clinically compelling strategies. So with that, thank you. And a lot of the work, as I mentioned earlier, is led by Dr. Nicole Gross and other key members of the Define team, and of course, other members of my lab who have contributed throughout this development, and then the funding sources for my lab. Thank you, and happy to answer any questions.
Thank you very much, Dr. Ho, for a fantastic talk. A reminder to everybody, you can use the Q&A function at the bottom of your screen to ask questions. We do also have some of the commercial and technical experts from the Enrich Biosystems team here today. So if you have questions about the TROVO platform, this is a great time to ask those questions as well. I'm going to go ahead and start us off with a question myself. Dr. Ho, you showed us a list of markers. These are a great example. What do you know about our ability to expand beyond that, or if we're interested in other biological contexts altogether?
Yeah, thank you for that question. The panel that I've shown you is just one mass cytometry panel, and right now, mass cytometry is the technique that we have applied, and so really the limitation is there, and we have about 40 to 50 markers within that, and so we have the chance to switch out. I mean, this is the reason why my lab, a lot of times we use mass cytometry for immune profiling, similar to flow, but there's no fluorescence overlap. So it really lends itself to ease of customization. And so you can have totally different panel. It really is not limited to the panel of antibodies that we use for myeloid phenotyping, obviously. So we can include, and depending on the context and the cell types that are you're interested. So for example, if you're interested in other biological contexts, like I've mentioned already, fibroblasts, it's such an important part of the tumor microenvironment. Or maybe it's not even a cancer context, so some other, context that needs to be studied. Then we can just develop a whole another antibody panel that's dedicated to phenotyping. And so, yeah, I think right now it's just one example, but the extension to other application is quite limitless.
So is mass cytometry the only platform that can be integrated with the TROVO system, or are there other analysis tools? And I'll start with you on that, Dr. Ho, and then maybe I'll open it up to the Enrich team as well to see if there's anything they'd like to add.
Yeah, so happy to expand on what I've alluded to earlier. So, when we were discussing as a team with Enrich Bio team, I think we were exploring the possibility beyond mass cytometry as well. It really is not limited to this. It just happens to be the technology that my lab has most familiarity with and expertise in. So we were able to develop an integration pretty easily. But you can imagine that any other single cell type of platform that can image a chamber slide. And there are many out there, really a ton out there, Koya, Lunaphore, all these other platforms have the ability to then phenotype and image these single cells. And in some ways, they have all different limits and different throughput. So we have to think about these considerations as well. But all of them allow you to do single cell phenotyping. And so because we're able to barcode every microwell and the information therein, we can then integrate across any other single cell methods. Now, a lot of the other methods I've alluded to just now are also not terminal. So I mentioned this, and what do I mean by that? Well, when we use mass cytometry, we have to ablate through the slide. So as the UV laser goes through and it ionizes all the cells into plumes of ions that can then push through at a per pixel level. Well, for other fluorescence microscopy or other high parameter microscopy methods, we don't need to destroy the cells. So you can imagine that in those cases, we can get the information and even go downstream and to collect these cells as Enrich Biosystems allows you to do and actually sequence certain wells that are particularly important. So yeah, I'll just leave it at that.
She or George, anything to add about other ways that other labs might be using the system, other analysis platforms they're using in tandem, or applications we're excited about?
Yeah, I mean, to me, we build TROVO to grow single cells as simply as growing a bulk culture. So people might not notice that the first control experiment that Dr. Ho with the T cells like can completely eliminate the tumor and in the presence of interferon gamma, and also the tumor can grow happily without all these killers. That's actually a bottleneck for single cell cultures because if you just make plastic or glass microwells, you're gonna have very poorly like nutrient and oxygen supply. So you're gonna have a necrotic core within each single cell, like single wells. Well, in our system, everything is made of hydrogel, so nutrients can pass through. So you don't have to optimize too much like from your bioculture. You can get almost the same readout if you do it using 96 wall plate or doing it using our like a tiny microwells, tiny hydrogel microwells. Again, based on that, you can use open-ended or terminal tools. Like I can easily think about instead of to calculate the like Dr. Ho is comparing top 50 and bottom 50 killers characterized by TROVO, and then compare a set of known or past candidate markers. You can just capture the top killers and capture the bottom killer and sign out for sequencing. You're gonna have a more open-ended result. So maybe you're gonna have more noise, but you're gonna have more sense to work on. So yeah, that's just how TROVO works. So we just handle the cell culture and the functional phenotype, and we try to make it open for other tools, other ideas. Yeah, that's a goal.
Got it. Thank you for adding that color. Let's turn the focus back to Dr. Ho's research program. Did you incorporate T cell proliferation into your phenotyping?
So yeah, that's something that we want to eventually get to. You know, right now the assay that's been set up is just in the context of cancer kill as the main readout. But I've also briefly mentioned this as a potential future application. So if you set it up in such a way where T cells are being activated with certain peptides, for example, and you want to capture that instead, and maybe the interaction with other cells in that process. We just have to set it up so that the fluorescence readout for each microwell provides the T cell activation. So, and then within the panel, we do have Ki-67 already. And so that's the phenotyping of T cells in whatever system or biological context, because my cytometry can phenotype whatever marker is part of the panel. Ki-67 being part of that can also get at T cell proliferation. So multiple ways to do this. You can imagine interferon gamma, TNF alpha, all these other readouts can be built into the mass cytometry panel as well.
So is that a future direction for you all? Are you going to be looking more into the T cell proliferation in the future?
Yes, we do have some ideas, you know, as part of the GI oncology program at Johns Hopkins, and our focus really having been in cancer immunology. We've also been developing new ways to immunize the patients to cancers, right? All these next generation vaccine platforms that are being developed. And so we have a lot of reasons and interest in utilizing this system to understand vaccine responses, for example.
So from what you understand today, do you expect there to be a lot of overlap between the tumor killing markers and the T cell proliferation markers? How do you imagine the interplay for those will sort of play out?
Yeah, I think the platform is flexible. It can be integrated in different ways to get the information that is most meaningful for the question at hand. So, for example, for a T cell activation readout, we may be able to screen their responses to various peptides at a very high throughput manner. And so and we want to be able to do this even with primary human samples. And so being able to get there, I think it's going to be a very powerful application in the future. And we're really trying to make that a reality.
So thinking about other labs that might be interested in getting this platform up and running for their own research, can you tell us a little bit about the experience of onboarding the TROVO technology?
Oh, of course. Yeah, absolutely. You know, this is not just because I'm on this webinar, but I thought it was very seamless. And I thought the whole team at Enrich Biosystems very supportive from the very beginning. And you know, I think what really made this such a productive experience so far is not only, yes, all the nuts and bolts were worked out very, very well. And so it was pretty easy to get it up and going. Really, by the way, we didn't have a ton of troubleshooting. Even when we had to develop this from scratch, this integration, it worked out pretty smoothly. It was really marked by the sense of collaboration. So this is what I think fostered our development of this method. And I just want to highlight that overall experience has been very positive.
Thank you so much for that insight. One of our audience members would like to know, have you tried to use the TROVO system to detect or measure mitochondrial function in T cells or myeloid cells?
That's really an excellent question. I think depending on the system, so for example, we've used the cancer cells in here that are expressing GFP as a readout. I've mentioned T cells, so if it was like an NFAT reporter, that could be used. Now, if you have a reporter for mitochondrial function, I believe that would be also easily achievable if that's the functional readout that you're interested in. If you want to be phenotyping mitochondrial function in the setting of other readouts, so cancer kill readout, but you want to also measure mitochondrial function, I'm sure you could do that too. If as long as there are antibody clones and a marker that you're particularly interested in, whether it's some sort of PDH or some enzyme, metabolic enzyme that could be rate limiting for mitochondrial function, I think that could also be incorporated very well into the readout because we have all these flow-based, cytometry-based methods to read these out already. And so just by showing how successful we were in being able to incorporate the cytometry phenotyping on top of the co-culture, I think that could be done with the mitochondrial readout as well.
Awesome. Thank you so much for that. And thank you all for coming today, for being an engaged audience. If you see on the screen here, we do have the URL for the Enrich Biosystems website. If you guys want to get in touch with their team or learn more, see what a demo looks like and how to set that up, you can do that there. And you can also fill out a contact us form and be in touch. So thank you so much again, Dr. Ho, for the great talk. Thank you everyone for coming and happy holidays.